I'm Jon Coopley. I'm Professor of Ocean Exploration at the University of Southampton in the UK. And I'd like to talk to you about hydrothermal vents, some of the geology that creates hydrothermal vents, and why they're very special habitats in the deep ocean. So hydrothermal vents are hot springs on the ocean floor. So think of them as like the geysers that you have in Iceland, but underwater. And instead of erupting steam, they erupt mineral-rich hot fluid. So we find most hydrothermal vents along the world's seafloor spreading centres. So that's the global mid-ocean ridge, where the plates of oceanic crust are separating as they ride on convection cells in the mantle. And also back arc spreading centres, which are slightly different, but basically another place where the ocean crust is being rifted apart by tectonic forces. So they're dotted along those global seafloor spreading centres. If we have a look at a cross-section through the mid-ocean ridge, it looks something like this. You've got maybe a couple of kilometres of water depth. The average depth of the crest of the mid-ocean ridge is about 2,500 metres. And then in the crust itself, there are layers of different types of rock. So the uppermost layer, the seabed surface at the mid-ocean ridge, is a layer of pillow basalts. And those are the lavas that have erupted from the rifting plates. And that's pretty permeable. Seawater can percolate down through those pillow lavas. And then beneath the pillow lavas, there's another layer that we call sheeted dikes. And those are the volcanic feeders for the pillow lavas. And that's also a layer that is permeable to seawater. And then beneath that, though, we get a change in the structure, and we have what are called crystalline gabbros. And those are not permeable to seawater. So what happens is seawater percolates down through the layer of pillow basalts, through the layer of sheeted dikes, and then it reaches that layer of crystalline gabbros. It can't go any further. And typically at that depth, that's about 2 kilometres below the seabed into the ocean crust, it's then encountering some sort of heat source, maybe a magma chamber that's beneath the mid-ocean ridge. So at that depth, it's now at high temperature, probably about 1,200 degrees C, and under high pressure and high temperature, that seawater reacts with the surrounding rocks. It loses some of the minerals that are typically dissolved in seawater, particularly magnesium minerals, which are some of the minerals that are washed off land as part of the water cycle. So they get deposited in the crust there. But it also leaches other minerals from the volcanic rocks. So it becomes enriched in a lot of minerals, a lot of dissolved metals, copper, iron, lead, and a lot of reduced compounds as well. So things like hydrogen sulfide, hydrogen, maybe some methane as well. So we have a hot fluid formed in this reaction zone deep in the crust. And because it's hot, it's buoyant, it's different to seawater, so we call it hydrothermal fluid now. It rises back up to the surface of the seafloor, erupts from it as the hydrothermal vents. So what we see at the hydrothermal vents, the classic sort of black smokers that we have, they are the end point of this circulation through the crust. Okay, this hydrothermal circulation. And it's very important for maintaining the geochemical balance of the ocean, because the minerals that are being washed off land are the ones being deposited in the crust, so they don't accumulate over time in seawater. Basically, it helps us to understand why the sea doesn't really get saltier and saltier over time. It's part of maintaining that geochemical balance. But in terms of biology, we have this hot mineral-rich fluid, which is enriched in particularly reducing compounds, things like hydrogen sulfide, which provide an energy source for chemosynthetic microbes. So these are microbes that can use reducing compounds as an energy source to synthesize organic molecules from inorganic carbon. So very similar to photosynthesis, but not driven by sunlight energy, driven by chemical energy. And those microbes in turn form the base of the food chain. They provide food for lots of animal species. And at hydrothermal vents, we get hugely abundant life compared with elsewhere in the deep ocean. And very steep physical and chemical gradients. So where the animals are thriving around hydrothermal vents, as you can see here, Actually, the temperature is probably maybe 20 degrees where the animals are most abundant. Where the fluid is coming out, erupting as that black smoke, actually just before it turns into black smoke and the minerals precipitate out, it can be more than 300 degrees. So we've got a huge range of temperatures over a scale of just a few meters. So very steep physical gradients like that, also very steep chemical gradients. The hydrothermal fluid is enriched in things like sulfide, it's anoxic, it's very different from seawater, it's mixing with seawater. And so we get a range of chemical conditions. And what we find is not only abundant animal species, but they live in a zonation around the hydrothermal vent. So you get some species that dominate very close to the source of the vent fluid, and then a little bit further away they're replaced by an abundance of another species, and then another species, another species. And it's a zonation, and it's actually very similar in scale to the kinds of zonations we're familiar with on rocky shores. We can walk 6 meters down a rocky beach and we will see a change in the dominant seaweeds and animals every few meters. It's very similar scale pattern of zonation because, again, very steep physical chemical gradients at hydrothermal vents. Now, hydrothermal vents are island-like habitats. So we have a cluster of these hot vent chimneys erupting the mineral-rich fluid, covering an area that's maybe a few hundred meters squared of the seafloor. And then it can be tens of kilometers, maybe hundreds of kilometers to the next cluster of active hydrothermal vents. We call each cluster a vent field. And so vent fields are dotted along the seafloor spreading centers like the mid-ocean ridge, and they're island-like. There's a cluster and then it's some distance to the next island habitat for hydrothermal vent species. And how far they're separated depends on the geology. So if we're somewhere like the Eastern Pacific, where the plates of the ocean crust are being pulled apart quite rapidly by tectonic forces, our hydrothermal vents are actually quite close together, maybe just tens of kilometers apart. If we go somewhere where the seafloor spreading of the plates is much slower, like the mid-Atlantic ridge, the hydrothermal vents are further apart, typically hundreds of kilometers from vent field to vent field. And each individual cluster of vents does not last forever in its hydrothermal activity. That flow of hot mineral-rich fluid will eventually naturally shut down at that particular vent field. And how long it lasts also depends on our geological setting. Fast-spreading ridges, like the East Pacific Rise, maybe activity at one site lasts for decades, and then it shuts down because maybe earthquakes rupture the plumbing of the fluid, or a lava flow comes in and covers the vent field. We've got a lot of activity at fast-spreading ridges. Slow-spreading ridges, like the mid-Atlantic ridge, vent fields last much longer in activity, probably active, and we know this from geochronology of sulfide minerals, active for thousands of years at a time. And often on slow-spreading ridges, switching on and off for a few thousand years at the same location. So quite different systems in terms of their dynamics. And the thing is that they are island-like habitats. So the species that we see thriving around them must have some way of dispersing from one vent field to another. Otherwise, they couldn't persist, they couldn't survive, because any individual vent field will not last forever. It will shut down, and then there will be no more chemosynthesis. You know, the species that depend on that food source will die out at that location. So what we see at the vents, the animals we see thriving there, is just one part of their life cycle. And it's this island-like system that makes hydrothermal vents a very good model system, if you like, for studying how species disperse and evolve in the deep ocean. So we might have a couple of hydrothermal vents quite close by to each other. We've got species at one, they're thriving, they're reproducing, and it's usually the larval stage in their life cycle that is how they achieve dispersal to another hydrothermal vent field, so that their population in space will persist across several vent fields. So we have this process, what metapopulation dynamics, this exchange of offspring up into the water column, carried by currents, reaching other vent fields. But sometimes that flow of larvae from one site to another will be interrupted. There might be a change in ocean currents. There might be some geological event that basically isolates a set of hydrothermal vents. And so where there was a population of species shared between sites, now we get them isolated. And then we get speciation taking place in isolation. And so we start to see different species in different regions. And that gives us what we call a biogeographic pattern. As we explore hydrothermal vents, we look at different regions of the world's ocean, we find different sets of species at the vents in any one region. It's like a regional species pool, and then we move to a different region, it's a different regional species pool colonizing all the vents in that area. And that's a biogeographic pattern. And it's like you can think of it as like a global jigsaw puzzle. And we've been spending the past few decades getting all the puzzle pieces. Going to new areas of the ocean where we know there are hydrothermal vents, but no one's seen the life, finding what species live there and piecing together our puzzle to understand it. So to give you some idea of how life at vents differs in different regions, this biogeographic pattern. Here we've got a map of vent biogeography. All the coloured pins that you can see, well, if they're the same colour, that means that they share the same pool of species in that region. That's one biogeographic province. Okay, where the pins are a different colour, it means there's a different set of species living at the vents in that region. So let's have a look at the blue pins on the map in the Eastern Pacific. This is one biogeographic province. Animals we typically find at hydrothermal vents in this province would be things like, well, right on the tops of the high temperature black smoker chimneys, we have these alvinellid polychaetes. Okay, so they're living in the hottest part of the vent environment. They're feeding on bacteria on their bodies. They live in U-shaped tubes on the chimney surfaces. They're in the innermost zone in our zonation of life in this biogeographic province, Eastern Pacific. A little bit further away from the source of vent fluids, we have these red-plumed tube worms in Riftia pachyptilla that you can see here. So this is an animal that's living with microbes, chemosynthetic microbes inside it in a bag-like organ in place of its gut. It provides them with what they need for chemosynthesis. It's able to harvest the microbes that are thriving inside its body. That's the kind of the next zone out from the vents. And then beyond that, we might have a bed of mussels. Again, dependent on chemosynthesis, actually with microbes associated with their gills that are providing them with nutrition. So that's just 3 zones, 3 different species that we get in that biogeographic province in the Eastern Pacific. If we jump now to the Atlantic and we have a look at the mid-Atlantic ridge and the vents that we find there, it's a completely different species. It's a different biogeographic province. So in that innermost zone around the black smoker chimneys, instead of alvinellid polychaetes, we have shrimp like these Rimicaris exoculata. So completely different animal, but still feeding on chemosynthetic bacteria growing on their bodies. Okay, so actually quite similar functionally to alvinellid polychaetes, though it's a completely different type of animal. Next zone out, where we had tube worms in the Eastern Pacific, we don't get tube worms at mid-Atlantic ridge vents. Instead, we have often these sort of fields of anemones in place of the tube worms. And then beyond that, we might occasionally get mussel beds, similar types of mussels, but a different species. Same genus, but a different species in the Atlantic. So different species, different biogeographic provinces. So the big question is, okay, as we go around the world and we get different puzzle pieces, we find different species in each province in this zonation, but what's driving the overall zonation? The animals are different each time, but what is the same? What's actually behind these patterns? Well, it all depends on the microbes with which the animals form partnerships. So in our innermost zone, whether it's Eastern Pacific alvinellid polychaetes or it's mid-Atlantic ridge Rimicaris shrimps, those are animals that are in partnership with epsilon proteobacteria growing on their bodies, and they tend to be feeding on those. And the same is true for some of the other provinces as well. It tends to be species feeding on epibionts, so bacteria growing on their surfaces, not inside them, and they're epsilon proteobacteria for a reason that we'll see in a moment. The next zone further out that we have a look at, where we get the tube worms in the Pacific and so on, that's different. That tends to be animals that are in partnerships with gamma proteobacteria. And often the gamma proteobacteria are endosymbionts. They're living inside the animal in organs inside them, like this trophosome bag-like organ in the Riftia tube worms. So partnerships with different microbes in the next zone out. So the question is, why do we get epsilon bacteria and their animal partners living close into the vent? Why is it gamma proteobacteria living further out from the vent? And it depends on the metabolism of these bacteria. Epsilon proteobacteria can use hydrogen as a chemical energy source, and they can oxidize it using sulfur or using oxygen. They can also oxidize sulfur using oxygen. Now, close into the vents, those are conditions where oxygen is less available. So here we have a microbe that doesn't necessarily need oxygen for its chemosynthesis, and hydrogen tends to be a little bit richer in vent fluid. So they can thrive really well close in because they can make use of a broader range of compounds for chemosynthesis. Further out, where we get the gamma proteobacteria, well, they can only do chemosynthesis through sulfur oxidation. They have two pathways for sulfur oxidation. They can't use hydrogen and they need oxygen for their chemosynthesis. So they can only thrive where oxygen is more available in that zone that's further out. But their metabolism is more efficient. It is more productive than the epsilons. So there, okay, they can only thrive a little bit further out, but they will outcompete the epsilons in that zone. So we have a zonation of our chemosynthetic microbes. And although the animal partner changes, that kind of geomicrobiological template is the same in every province. It's just the animal partners differ. Okay, so that's the kind of thing we get from exploring biogeography. Initially, it looks confusing because it's different animals in different regions all the time. But when we look at the organism, not just as the animal, but the partnership between animal and microbe, then we can understand the pattern of life. So beyond the species that have these intimate partnerships with chemosynthetic microbes, in our zonation, the next zone out, we get dominance of filter feeders, so things like stalked barnacles, those sorts of organisms. That can extend over tens of meters. We often get maybe some predators and scavengers in that zone as well. And then eventually, our little island of life is surrounded by a halo of animals that we're familiar with from other parts of the deep sea. They're not unique to hydrothermal vent habitats, but they're present around hydrothermal vents at greater abundance than we're used to seeing them because they are making use of the productivity of the hydrothermal vent that's sort of still being exported. So we get this halo, as we call it, of non-vent fauna at elevated abundance. And that gradually, over hundreds of meters, fades into the normal deep sea background fauna. And that's our island of life at a hydrothermal vent. So the important thing about hydrothermal vents are they're island-like habitats, which means we end up with biogeographies. They are ephemeral as well. So all the species we see thriving around them have to have some way of dispersing from habitat patch to habitat patch.